Imagine plugging a silicon microchip into a machine, running an electrical current through water, and watching it print the actual building blocks of life. It sounds like science fiction, but researchers at Harvard University have recently accomplished this in a lab. To understand why this matters, we have to look at how modern science operates. Creating custom DNA sequences from scratch is how researchers engineer bacteria to clean up waste, design targeted therapies, and develop modern vaccines. It is a necessary foundational component of almost every biotech laboratory in the world. Biotechnology usually feels like a vision of the future, yet the process of actually manufacturing this biological code remains surprisingly tethered to the past. Right now, the entire biotechnology sector relies on a process called phosphoramidite chemistry to write DNA. It is a method that has remained the industry standard for over 40 years. While it works reliably, it requires massive, specialized factory equipment. And more importantly, it depends on pumping hazardous chemicals and harsh organic solvents through those machines to link the DNA building blocks together. Handling and disposing of those toxic byproducts makes creating synthetic DNA expensive, heavily centralized, and an ongoing environmental challenge. We are essentially writing the advanced code of biology on a chemical typewriter. Advancing medical and biological engineering requires moving past hazardous fluids and finding a way to control this chemistry through electronic circuits. The Harvard team bypassed the harsh chemicals by asking if electricity alone could control the reaction. To test this, they designed a specialized CMOS semiconductor chip. CMOS is the family of technology used to build the processors inside your laptop and the circuit boards in your smartphone. But this specific hardware was originally engineered to sit inside the brain and record the faint electrical chatter of thousands of individual firing neurons. The researchers realized that the extreme electrical sensitivity required to listen to brain cells could be reversed. Instead of listening, they could send precise electrical pulses out to control microscopic chemical reactions in a liquid solution. This crossover between neuroscience and genetics proved that standard silicon hardware could orchestrate the assembly of genetic material. The surface of their chip is divided into 64 programmable synthesis sites. Surrounding each of these tiny locations are two microscopic circular electrodes arranged like an inner and an outer ring. When an electric current runs through the inner ring, it zaps water molecules to generate protons. This creates a tiny, highly localized droplet of acid, which removes a protective block from the growing strand of DNA so the next genetic letter can be attached. The problem with generating acid in a liquid environment is that it doesn't stay put. If those protons drift away from their specific site, they will contaminate the surrounding areas and ruin the other 63 DNA sequences being built nearby. To solve this, the outer ring acts as an electronic vacuum. It instantly absorbs any excess protons, trapping the acid strictly within its microscopic borders. Because they could confine the acid, the team successfully printed 64 completely distinct DNA sequences at the same time on a single chip. By turning every point on that grid into an isolated chemistry lab, they showed that DNA manufacturing no longer requires massive physical infrastructure. But this hardware is not ready to replace traditional manufacturing just yet. The DNA strands printed in this study were only about 39 bases long. Real genes often require hundreds or even thousands of bases linked together in a continuous chain. The bottleneck isn't the electronics, but the chemistry. Reactive molecules created during the process can still move between sites if they are packed too closely together, which limits how dense these synthesis locations can be. Even with those limitations, the team demonstrated the potential of the system by encoding a 169-byte text message directly into the synthesized DNA strands. Using synthetic DNA to store digital text provides a path toward one of the highest density storage media ever proposed, one where information is stored in molecules rather than magnetism. As researchers overcome these chemical limits, this process could scale using the exact manufacturing lines that stamp out computer parts, making DNA production cleaner, faster. Widespread access to high-quality synthetic DNA could accelerate work in synthetic biology, personalized medical treatments, and speed up new gene therapies. What do you think? Will a standard desktop-sized DNA printer eventually become a common piece of equipment in research labs and hospitals?